I saw the work that was done with the peripuberty with the rat, I started thinking, well, it would be really tasteful if we evolved so that during this period of really marked biological change of puberty and marked hormonal regulatory change of puberty, maybe that helped open up the system again to sample how harsh or benign is my context and to sort of adjust systems, a stress system accordingly, right before you reach the time when you're reproductively ready to have offspring, right? Sort of a reasonable time to readjust.
Welcome to The Stress Puzzle, where we explore the latest in stress science and consider how the science may translate to our daily lives or where we might have missing pieces for actually making that connection. I'm your host, Ryan Brown, and I'm a social health psychologist working with the Stress Measurement Network, which is a team funded by the National Institute on Aging and includes internationally recognized stress experts from UCSF, UCLA, and Yale. In this episode, I speak with Dr. Gunnar about the calibration and recalibration of human stress systems. Dr. Gunnar has explored these ideas in the context of early adversity and with a focus on the importance of puberty as a window for recalibration. We're joined today by Dr. Gunnar, who is a Regents Professor and Distinguished McKnight University Professor at the University of Minnesota. Her doctoral training was in developmental psychology at Stanford University, and she then completed her postdoctoral fellowship in psychoneuroendocrinology at Stanford Medical School. Since then, she's built a remarkable career studying how stress biology affects neurobehavioral development and all the processes that help children regulate stress hormones. Dr. Gunnar is a fellow of the National Academy of Sciences and has numerous lifetime achievement awards across many societies and disciplines, including the Association for Psychological Science and the International Society for Psychoneuroendocrinology. I especially admire her interdisciplinary approach to addressing questions of early life adversity, stress biology, and developmental timing. So welcome, Dr. Gunnar. We're so happy you could join us here on The Stress Puzzle.
It's wonderful to be here with you today.
I was hoping you could start us off by helping listeners to sort of conceptualize what calibration of stress response systems really means in the context of developmental psychoneuroendocrinology.
Okay, maybe it's better to start by thinking about the fact that our bodies have to adjust themselves to where we're going to grow up and where we're going to live. And throughout evolution, a number of mechanisms have developed to be able to do that. There's even evidence that how the size of our sweat glands are influenced by how hot it is in the first 6 to 12 months of our lives. And that is, you calibrate. If I'm going to be living in a really hot climate, I better be able to sweat more than if I'm living in a cold climate. Calibration of the stress system is similar to that. It's the idea that if the environment you have been born into is full of challenges, perhaps from famines, perhaps from extreme cold or extreme heat, or from a lot of various kinds of adversity. A lot of people die, et cetera, et cetera. You need to calibrate your systems to your stress systems to be able to manage that degree of challenge, that harshness. However, if you're born into a pretty benign setting where there's plenty of food and everything is wonderful and people aren't dying all around you, then your systems get calibrated to that more benign kind of context. So the calibration is calibrating according to the demands that you are likely to experience during your lifetime. But we have a very long lifetime as compared to a rat or some other much shorter lived species. A number of us have wondered whether there are periods of recalibration during the course of development.
Yeah, and so it sounds like this idea of calibration more broadly is really well understood and accepted, and adding in sort of the social environment as part of that calibration system is a lot of what is really unique about this interdisciplinary advantage that we kind of can see. And you focused a lot on the hypothalamic pituitary adrenal axis, especially during puberty. And so I'm curious if you could talk us through sort of what's special about puberty, especially for stress systems.
Well, I'm going to take you back a little bit. The calibration of the HPA axis and the importance of social experiences is certainly not my contribution to science. We've known about it since Gig Levine in the 1950s and early 60s recognized that the kind of stimulation that the mother rat provides for the infant rat calibrates that infant rat's HPA axis reactivity and proneness to anxiety. my contribution was to show that, oh yeah, it also is true in us as humans. And the early period is a very important period, the first few years of life, 2, maybe 3 years of life, very important for that initial calibration. Puberty is this amazing period of time when there's vast hormonal changes, and others have began before me to notice that reactivity of the HPA axis actually increased over the course of puberty for a period of time, probably along with all of the other steroid hormone changes that are going on at puberty that have to do with maturation of our capacity to reproduce. So, and there was some rat work suggesting that that period of time was also another sensitive period, in that stressors that confronted the rat during this peripuberty, around puberty, peripuberty means around puberty, period, would have longer lasting effects than the same stressors encountered in the mature rat.
Yeah, and it really ties well to something I really appreciate about your research. And I think we want to highlight in this podcast is how to do this work. Largely, you really do need to be able to go back and forth, or it helps a lot going from human models to animal models to maximize the different ways that we might consider how early life adversity or any other factor that we consider may bridge to later life health and stress responses. So I guess I'm curious if you could talk about how you've approached working with both animal and human model research to support the hypotheses that you have.
Well, one of the challenges of working with humans is there are many things you cannot do experimentally that are done to kids every day. In the rodent work, and there are things you can do that are under controlled conditions to really test causal mechanisms. Really tough. We can randomly assign kids to improve their lives, but we cannot randomly assign kids to adversity, and never would want to.
Of course.
The other thing, no, we would not want to, and which is why I say it happens to kids all the time, and worse than we would ever do if it was experimental. But and we have to remember that it's not that children don't experience real awefulisms. The animal models also allow us to get at mechanism. With kids, especially with young kids, we can't even really draw blood in order to find out what's going on in their bodies. We're pretty well stuck with really non-invasive measures. Of course, we can image the brain, but we aren't going to go in and dissect it. So to really get at mechanisms and where things are happening and so forth, you need the animal models. But nature never put an animal on Earth to model a human.
Mm-hmm.
Not done that way. So we call them animal models, but they're really, in many respects, to answer the question of how did this species evolve to function in its ecological niche?
Mm-hmm.
What is the story for that animal? Okay, if I understand the story for that animal and another, this is comparative, like not just working with an animal model, can I begin to ask questions about what the human chapter to that mammalian story might look like? Having an understanding of how those chapters were written for other mammals for whom we can get at mechanism and control experiences more. Does that make sense?
Yeah,
I hate,
absolutely.
I understand we use the term animal model, but I tell my students, it's not really a model.
Right. It's a completely different experience and biological system, but being able to get at those mechanisms is how else would we do it? The wealth of knowledge that we've gotten from different species. I do a lot more in adults and think about attachment and adult relationships. So we think of prairie voles, but again, huge differences in prairie vole relationships than human relationships.
Huge differences. Sarah Hrdy, I'm sure you know who that is, HRDY.
Mm-hmm.
Fabulous anthropologist, biologist. As she once said in one of her books, that Mother Nature rarely buys anything new. She uses what's in her larder to
Mm-hmm.
solve the problem. So that is a fact. There is continuity in evolution. And so it's not like unlikely things are done wholly differently, but it always worries me when we think, oh, so nature did it just exactly the same. Probably not.
And
It's a different.
especially when we're thinking, lifespan type questions, just the sheer difference in lifespan across these different animals. So I'm curious how you've addressed that or dealt with that, like the lifespan differences.
Well, it's precisely why I began to wonder whether I saw the work that was done with the peripuberty with the rat. I started thinking, well, it would be really tasteful if we evolved so that during this period of really marked biological change of puberty and marked hormonal regulatory change of puberty, maybe that helped open up the system again to sample how harsh or benign is my context and to sort of adjust systems, a stress system accordingly, right before you reach the time when you're reproductively ready to have offspring, right? Sort of a reasonable time to readjust. So that's what led me to think about that. There's other periods. I have a wonderful student, Marianne Howland, who's really interested in the possibility that pregnancy and for both the woman and possibly for the man, as he gets really involved in taking care of his babies, might be a period that opens up the system again for a bit of recalibration.
Absolutely. I'm excited to see all of that work. The incredible reorganization of life around that time and biological recalibration as well. Just diving into your research a little bit more specifically, as you've been touching on, I really admire the ways that your creativity in testing your pubertal recalibration hypothesis. And so I was hoping you could talk us through what you were really looking for in a sample to test this theory and, what you found when you did find that sample.
Well, you have to understand that I had been spending quite a bit of time using kids adopted from orphanages as a model of early adversity. It's a very important model for us to understand, both for the kids themselves, of course. These kids were not there just to help me have a good career. But one of the things that was important about them is they started out under conditions, most of them, that were definitely harsh. They were not what we evolved to anticipate. Biologically, we evolved to be cared for by one or a few people, cared for by a whole mixture of people with no identifiable attachment figure, and on and on and on in atmospheres. It's very, very difficult when you put 20 babies together not to have a lot of germs and guck. Babies are dirty. They are, especially when you put them all together. It's hard to maintain that low. So the system would be challenged both by pathogens and parasites and, lack of responsive care and on and on and on. Okay, we got that. Now, unfortunately, for most people, the conditions of your early life are fairly predictive of the conditions of later life, right? Dreck tends to follow Dreck, and good things tend to follow good things. Don't have a dramatic reshuffling of our social classes. There is some, but it's not very dramatic. So I wanted to test really whether there was an early calibration period for the stress system. I needed kids who experience radical change in conditions. And that is absolutely true for kids adopted internationally from orphanages. It's not that life becomes perfectly sweet. Many of them are being adopted into homes with white parents when they're children of color and are moving into the US or other countries where they're then also discriminated against. So that everything's not perfect. But in terms of radical change in moving to high resource homes with people who really want to be responsive to you and care for you and raise you and do all the great things for you is a radical change. And so I initially sought this group as a way of testing the initial calibration of the stress systems and whether it would revamp as soon as they got to goodness. And it doesn't. And that was what blew me away. It did blow me away. I really expected more flexibility in the system. And instead, what I found was that there's a downregulation. There's a hypofunctioning of the adrenal gland. I don't know about the rest of the system. I can't find out about that in the kids who are adopted internationally. And the degree of hypofunctioning is associated with problems with ADHD and peer relations and et cetera, discriminant friendliness and problems in attachment. So then I began to wonder, well, does puberty give an opportunity for the system to reawaken and recalibrate? Or are these kids going to be hypofunctioning the rest of their life? Yeah. So the question grew out of my original seeking of this sample to understand the initial calibration, curiosity about, well, is it going to be this way forever, or will puberty open another window?
Right. It really does have such incredible implications for policy. The difference of if that initial calibration is where you're at versus if you have this window of, more flexibility and reorganization that's possible. I mean, the profound difference in how much I would think policymakers would care, right, about improving conditions and things like that. So I just want to take a step back and say how important that contribution itself is.
Yeah, but then I went on and I said, well, okay, now you've gone from driving a Volkswagen to driving a Ferrari.
Mhm.
Now you've got a stress system that is reactive. Does that mean all this other wonderful recalibration is happening and the fact that you had emotional problems are all going to recalibrate and everything's going to be sweetness and light? And the answer to that is no. Now that you're driving a Ferrari, your brain was developed to expect a Volkswagen.
Mhm.
So we're actually seeing that the heightened reactivity with this calibration is predictive of increased internalizing issues.
And so this is sort of the link here of how behaviors kind of adjust or may be affected by this timing.
Yeah. I mean, the bottom line is there's no free lunch. If we want individuals to function well, we need to take care of them from the get-go. It's not that they're not going to, none of them are going to be able to, but on a population health level, taking care of making sure that life is good for kids from conception on is a pretty good plan, and that we can work at repairing problems. And in fact, man, internationally adopted kids, they show incredible improvements in functioning. I mean, it's not like, gee, it has no effect. No, many of them just show these massive, incredible rebounds in language and cognition and growth, and they still struggle with problems. I mean, they're doing incredibly better than were they to stay in the institution. Mm-hmm. But this period of recalibration of the stress system may raise additional challenges. And I think what we need to understand is whether providing additional support and training in emotion regulation. You may or may not remember when you went through puberty as a girl.
Probably blocked it out largely.
There was this crazy thing called cycling. And your emotions sort of that didn't have this cycling issue, it began to have this cycling issue, right? And you go through periods of wondering why you wanted to cry. And then you would, of course, go out and find a good reason and get really upset until you figured out that, oh, in a few days, my period's going to start. And this is just, a little mild bit of PMS. And so that's learning how to live with your new hormonal self. Does that make sense?
Absolutely.
So I've been wondering with this recalibration issue, you don't get the same kind of emotive feedback from cortisol that you do from estrogen and progesterone and all those lovely things. But there's a change in the way the body is working. And I've wondered if whether helping kids understand that they're going through this change process, or perhaps, especially because they're going through puberty at the same time, understanding that whole change in biology as not necessarily meaning anything emotionally. It's just your body's adjusting, right? Don't get all wrapped up in trying to understand why you're upset. Because it's just your body. It's going through its thing. Take a nap.
It reminds me of, if you're someone who doesn't drink a lot of coffee and then you have a day with more coffee and then maybe it's a more stressful day, suddenly your heart is racing. And how do you interpret that, faster heart rate there? Is it, I'm panicked and I will not be able to perform whatsoever? Or can you take the step back and be like, this is a biological response
caffeine.
And this isn't,
It's caffeine.
Yeah, it's just caffeine. No,
yeah,
that's a great point.
Yeah, I think there's a lot of that. Then I don't do intervention work. So I don't know if anyone's done that kind of training with adolescents to just understand, your body's going to be a little weird for a while. And it's going to send weird signals. Don't worry about it. It's fine. Just wait a day. Just wait a day and see what tomorrow brings. I don't know if they've done any of
it. It makes me wonder if there's something in like the PMDD world in line with that of just more difficulties with cycling that, persists beyond puberty. Anyway.
Well, some of this, I just, it's just theirs is probably not quite as predictable.
Right. And, something I found really interesting in thinking about your career was how, a lot of times in the stress world, we think okay, the way past stress might be affecting us in the future is by making us really react more strongly or have this hyper responsivity. But your work and others really highlights that this isn't always the case, and maybe not even the most likely case, especially for cortisol, and that we might really expect more hyper responsivity in response to early life stress. And I was curious if you could just speak to this distinction and if it was surprising to you and sort of how you might have navigated that, publishing those findings when that wasn't necessarily the dominant view in psychoneuroendocrinology at the time.
Yeah, well, poor Rachel Yehuda. I guess I shouldn't say poor Rachel, but at the time when she found it, it was poor. A wonderful neuroscientist, and maybe a name you know, who has studied PTSD. And when she was a young scientist, she was working on activity of the HPA axis and PTSD. And everybody and their dog, I mean, it was about 10 years before we began to get our findings. Everybody and their dog absolutely expected that people with PTSD would have elevated court. And she was finding low court. You know, all the silverbacks, because it was mostly males at that time, you know, were poo-pooing her, finding, oh, nice little girl, you've got that wrong. Well, she was absolutely right. Then we came along, and once we had salivary cortisol measures, the field, and people could leave the lab and actually have people sample as they went about their everyday lives. Weird stuff began to happen, and people like Dirk Hellhammer and others who were getting, and Clemens, but especially Dirk here for adults and biorhythms, began to notice that not everybody had a high morning and low evening. There were these people who had low and low. I mean, and not, there were these weirdos. And they wrote, he and Christine Heim, I think was the first paper, wrote the first paper on the possibility that hypocortisolism was a real phenomenon in humans, and perhaps associated with problems like chronic fatigue, et cetera, et cetera. It's not clear how associated it is with those problems, but that was their first paper. So when Millie Vasquez and I observed this, kids exposed to significant early adversity, and we wrote our paper, we were not the lone people out in the wilderness saying, whoa, maybe human beings go sort of hypo under chronic stress. And more of those findings began to come in. There are chronic depression is associated with slightly elevated morning cortisol and elevated evening. So a sort of a tend towards higher court, but a lot of other adversity is not. Mm-hmm. Or, people who are experience the same adversity and don't get depressed. Christine Heim has shown be hypofunctioning. But it was, the push was so hard to believe that it had to be sort of a prelude to hyper, that then there was the expectation that those people would become hyper reactive. Maybe not. There's a lot more evidence that in humans. Now, the question is why and where? Is it that your stress system, your central stress system has become hypofunctioning? Or is this down regulation happening more in the periphery? And we have data that at least raises the possibility that it might even be at the adrenal level. And the brain is, the brain is going on being hyper stress reactive, but chronic elevations in court really rough on the body. And so tend to be followed by mechanisms like get it back down. And there's old, very nice data that if in rodents, not in kids, if you inject corticosterone into the ventricles, so it's going throughout the brain, it tends to upregulate corticotropin releasing hormone in the amygdala and maybe downregulate it in the hypothalamus, like it unhooks the different parts of the stress system.
Oh, fascinating. Yeah.
Absolutely fascinating. Nature doesn't like to be put in a box. And so if nature needs to have us become hypervigilant, pay attention, there's chronic threat, but if we run really high levels of the peripheral hormones, it's going to, wear and tear us out way too fast. We'll have Cushing's and other things. Downregulate at the periphery, but keep the brain going. And then maybe there's also some possibility that unhooking, which may save us initially, also may make it hard to ever ramp down what's going on in the brain, because elevations in cortisol are part of turning off that central system as well.
Fascinating. And, I really appreciate that you touched on how measurement really played a role in that. Because I wonder what that would have looked like if we didn't have those salivary cortisol measures at the same time to really be able to, show this out in the field. So I'm curious what you view as like biggest innovations in stress measurement, whether that's on the biological side or psychological side, and what we might need to push the field forward in your view now.
Yeah, the hair cortisol has been really cool because it allows us to look at sort of the calendar of impacts and many other things than just cortisol. But it doesn't tell us about what happens in daily rhythms. So we need the capacity to also look at the daily rhythms that can get so bollocksed up under chronic stress. Everybody and their dog would love to be able to measure cortisol with just like a dipstick or something, or to be able to spit in something and 10 seconds later know what the cortisol level is. And there's been a lot of attempts to go and create that technology. It's likely to happen at some point. And that sort of immediate feedback would be potentially very helpful. You could imagine designing studies, biofeedback kind of studies to do with that. But we already can do that with heart rate and other things as well. The AI is likely to be critically important because the body responds in patterns and not just in one system at a time. So while all the systems talk to each other in different individuals of different genetics, the way they talk to each other and the sensitivity and the patterns that emerge are going to be somewhat unique. And the prediction, heterogeneity is something we really, really need to understand. And we can't really understand it when we go with just one measure at a time, or even two, or even three. And then as we get way into all the genomics, and we need AI and machine learning to help us put all that in.
No, you definitely touched on one of my biggest kind of questions or concerns around how we're just how well can we link these systems up. It always stands out to me that when we're isolating these systems and finding important findings within each system, just the how does it all connect together and bringing in genetics to that as well.
Well, obviously there are as many different patterns as there are people, but that is if you're going to predict every system perfectly related to every other system perfectly. As you loosen that up a bit, you begin to find groups that are operating sort of alike. And that's probably where some sweet spot where sort of alikes are predictive of certain, different patterns of outcomes are driven by different inputs. In doing that, one of the challenges we have, do we go to Dust Bowl empiricism? Like, we don't care that we understand something about biology. Just throw them all in a pot and let artificial intelligence sort it out. Or do we guide the work that is part of your answer to that, of,
Right.
Looking with some understanding of gene networks, of understanding of sort of how systems And I think my gut goes biologically informed AI would be probably what we want to do. But right now we're sort of getting really happy with just straightforward machine learning and seeing what it turns out.
Right. Right. Yeah, it'll be very interesting to see best practices moving forward with AI and machine learning and just the vast power that we're getting from these tools.
Mm-hmm, mm-hmm.
So I wonder if you just sort of take a step back on your remarkable career, if there's a particular finding that feels the most meaningful to you, either because of the importance of the results or something about the research process itself.
Yeah, my favorite finding that really changed my thinking about all of this was when I was finally released from the newborn nursery. I'll explain that in a second. Because I could move from blood to saliva. The salivary assays were working well enough, and I imported them into and was able to do them at the university I was at. Not me, the medical lab could do them. So initially, since I needed blood, I did work with, and I wanted healthy babies. I was confined to the newborn nursery because you could take extra drops of blood when they did the metabolic screening exams, the little heel sticks. And then I hired a nurse, and we would take the blood for those. So the phlebotomist didn't have to come, or whoever didn't have to come in, and we would organize that sampling around questions like what was the response to circumcision, all the rest of the things I did in the nursery. I was doing all that while the salivary assays were really coming online. Once I had those, I was like, yes, I can go out and I can look at the development of individual differences in stress reactivity. And the first study we did, and temperament, temperament, stress reactivity. Those were that linkage was what I was interested in. And we did a whole set of assessments of temperament using the Louisville temperament measure at 9 months of age. And I was going to predict stress reactivity to separation at 12 months of age. At 9 months, I was getting reactions just to the temperament assessment. And at 12 months, holy mother, the kids didn't respond. And I'm like, wait a minute, separation from the parent is like the biggest stressor we can use in a non-human primate. It's like big. And so that was when I called up Gig Levine, my former PhD mentor, and said that the adrenal's not doing what it's supposed to. And he sort of laughed and he says, it's doing what it's supposed to. You just don't understand it. I said, no, you're absolutely right. And you're not much help.
What a great and classic response.
Really could have knocked his head off. But that's led me to say, well, what in heaven's name is happening here? And at 9 months, the attachment system wasn't really, they were merging into it, but at 12 months, they were pretty securely attached. And you're not nasty to babies. You don't keep the parent away. First of all, I also started thinking about the difference between separating a mother monkey from her infant, and she does not like that idea. And so you are battling. Mother screaming and yelling, don't take my baby, and you're taking the baby. That's not what happens in humans. You say, Mom, would you go out of the room? And she says, yes, of course. So it wasn't the same degree of stress, I didn't think. And I began to wonder, I said, I knew the literature on social regulation of stress. There was some literature there. I thought, well, maybe the attachment system, Bowlby said it would be a buffer. And maybe it really is. And so then we did the studies that really showed the buffering effect, the powerful buffering effect of a secure attachment relationship. I also then began to think, well, it would make very little sense if every time a baby got upset, they racked their HPA axis up to the sky. But also in non-human, in rodents, there's this period of stress hyporesponsivity. But our system is much more mature at birth, so it's unlikely to be within the system. But even in the rodent, that capacity to remain high, to not react, is maintained by the mom licking and grooming the baby. And you take those things away. You take away milk into the gut and the licking of the anus, which we don't do, but they do. And the system becomes reactive. So even in the rodent, this immature period is being maintained by signals from the mother. So let's leap forward to humans. Much more mature system does react earlier in the first year, but now we've got a kid who can locomote, can go out and get itself into trouble, can cry and return to the mother figure. And maybe as long as the mothering figure is responsive and this attachment is secure, the crying is just a good signal to the attachment figure that I need help. Mm-hmm. And if help comes, well, why do I have to kick off my system? My mothering figure can elevate his or her stress system, which is what happens. Mm-hmm. So down that path and design a set of studies that would show that, that's what happens.
Gorgeous. The social buffering and distress regulation pieces, so close to my heart. So I love that that's what you identified there.
And Mike Hennessy has a beautiful paper. He's a guinea pig guy, was also trained by Gig Levine, on how social buffering appears for whom and at what stage of development and with whom in different species. It's a really lovely paper. Mm-hmm.
Amazing. We will definitely link that in the show notes so folks can check that out as well. Thank you so much for joining us today, Dr. Gunnar. I really appreciate everything you've shared today, and it's just been a delight to get to chat with you.
Well, it's been fun chatting with you, Ryan, too. Take care.
Thanks for tuning in to this episode of The Stress Puzzle. We'd love to hear your thoughts and feedback on any issues we've covered today. You can email us at stresspuzzlepod@gmail.com. And you can also send requests for topics or guests for future episodes. The best way you can support the show is by leaving 5-star reviews wherever you listen to our podcast and sharing with your friends or your collaborators. And until next time, we're wishing you good stress and opportunities for rest.